Today's topic is DNA replication errors by Hilah Torres in Dr. John Wool's class at Grand Canyon University for Cellular and Molecular Biology. The topic and objectives for today's lesson include the topic goes over the basics behind scientific discoveries of DNA and DNA replication in the last century. The lesson provides an overview of how DNA replication should occur and what happens when errors occur. The objective is for students to understand the history of DNA discovery, how DNA replication works, and to understand how errors can occur and what the outcome is when they are unable to be fixed. The importance of the concept here is that students will understand what can happen when DNA replication errors can occur. Worst case scenario, damage to the cell is done, or cancer can be an outcome. As our society moves to understand how and why cancer occurs, we can note that DNA replication errors happen constantly and that this is a natural occurrence. Students should see the importance of learning from history and be encouraged to move forward in continuing research on this topic. Today's audience is the presentation to undergraduate students who have a very basic background in high school biology. And the important part is that students would have a basic understanding of the vocabulary and terminology in this lesson to be able to keep up with the concepts that we are discussing. So I would definitely make sure to go over that with them. Replication of damaged and undamaged DNA. Adolescents learn at a young age that they are composed of genes passed down from both parents. At an even younger age, children notice similarities they may have from family members, and as they grow older, they may identify further with a relative that they look like. The simple basis of noticing physical appearances has much deeper roots, and we will discuss the derivatives of that today. All forms of life need a way to store, retrieve, and interpret genetic codes that make up a living organism. In this case, a human being. In the early 20th century, scientists began to discover genetics and how the information in genes is stored in the body, more specifically in the cells. From there, it's copied and then transmitted to daughter cells. The most fascinating part is that DNA replicates billions of times in an organism's lifetime and makes it out almost the same from the start, hardly changed. Discovering DNA, or deoxyribonucleic acid, is a fairly recent event in scientific discoveries. In 1953, James Watson and Francis Crick learned about how DNA is transmitted from cell to cell and how they contain codes that give instructions. Every cell on Earth contains DNA. Their scientific discovery is now named the Watson-Crick model for DNA. Prior to the discovery of DNA, scientists discovered chromosomes, which contain both DNA and other proteins called histones. Genes are what carry the hereditary information that is replicated and passed on to daughter cells when a cell divides. When looking at DNA, it's important to recognize that there is a specific sequence of coding that must take place to accurately replicate. Different organisms can have different nucleotide sequences. When there is a complete set of information in DNA, that is referred to as a genome. An astonishing fact is that a complete sequence of a human genome could fill nearly 1,000 textbook-sized books. And all of that is encrypted in our cells. Going back to DNA replication, though, it is important to recognize that this nearly perfect process is only that, nearly perfect. Sometimes mistakes can happen, errors occur that cause mutations. DNA replicates inside of the cell's nucleus, if it is a eukaryotic organism. Because prokaryotic organisms lack a nucleus, their DNA replicates in a different way, but we're not talking about that today. This lesson is focused on DNA replication of eukaryotic organisms that do have a nucleus in the cell. DNA replication usually happens during interphase. This is when cells grow and carry out specific processes and replicate DNA. The time DNA replication would not occur would be during mitosis, the time when cells divide. Humans have nearly 37 trillion cells inside of the body. One cell can be damaged tens of thousands of times per day. Over the span of a lifetime, with the number of cells in the body, that could potentially mean a quintillion DNA errors. This damage can cause serious problems, unfortunately, like cancer, because DNA gives your cells the instructions they need for protein synthesis. There are a variety of errors that can occur. DNA has building blocks called nucleotides. When nucleotides are damaged, DNA strands can be mismatched when paired up, and this is where the mutations are caused. DNA replication can occur when it shouldn't, and even sections of the DNA can get mixed up. Luckily, human body cells are equipped most of the time to handle these situations. Repair pathways in cells rely heavily on specialized enzymes. Now, these enzymes are specialized just like the cells in signal transduction. These enzymes are specialized to where different ones will respond to different types of damage. You can think of these as you would the difference between plumbers and electricians. DNA strands either run 5' to 3' or 3' to 5'. The enzyme DNA polymerase, for example, will only work to replicate DNA 5' to 3'. That is how specific these enzymes can be. To exemplify further, a common error is base mismatches. Each nucleotide contains a base. During DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to match each template strand. For example, the paired bases could be quinine with cytosine and adenine with thymine. Every so often, a mistake can occur. The enzyme can recognize this immediately and can fix it by taking off some of the impaired nucleotides and then replacing them with complete and unharmed nucleotides. These enzymes are very thorough with the work they do, but again, mistakes can happen. To ensure the process is going the way it should be, and to double check their work, another set of enzymes come through to sort of inspect the process. The same thing occurs here if they identify a mismatch. They can simply replace the incorrect nucleotide after taking out the error. The term is naturally called mismatch repair. This process seems nearly foolproof, and it definitely can be when considering how helpful the second set of eyes can be. However, DNA can become damaged after it has been replicated as well. Chemical changes can damage nucleotides. A common example is environmental exposure. It can come from a toxic chemical compound that is inhaled, such as breathing in pollution in the air or cigarette smoke. It can also be a naturally occurring compound found inside the body, like with hydrogen peroxide. In these cases, there are specialized enzymes that can come up to help repair the damage. Base excision repair is when a damaged base is cut out by an enzyme. Other specialized enzymes come in to replace the damaged base with a new one. UV light damage is harder to fix and can even damage the DNA shape. It can also cause two adjacent nucleotides to stick together, which further damages the DNA double helix shape. The term coined where proteins fix this damage is called nucleotide excision repair. It is when a team of proteins remove longer strands of about 24 nucleotides and replaces them with new ones that are completely undamaged. Double-strand breaks are the most dangerous, as they can lead to cellular death. The two main ways these are repaired are with homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of DNA that is comparable to what was damaged as an outline. We kind of trace it or look at it as an example. Enzymes come in and interlace what is damaged and undamaged and get them working so that they may get two complete double-stranded sections. Non-homologous is different because they don't use undamaged DNA as a template. Rather, the enzymes take out some of the nucleotides and simply fuses the ends back together as if the enzymes are welders. The latter of the two techniques, the non-homologous end joining, is not as accurate, as some problems can occur from the binding back together. But it's still a technique used and is proven to be helpful when sister DNA is not available to be the example. Now, most of the time, it is important for DNA to stay the same. However, mutations don't always lead to disease and cancer. They can actually be beneficial for a species, as some mutations can allow them to evolve to their ever-changing environment. For example, mutations have occurred in populations of people to build up an antibiotic resistance to bacteria. DNA mutations are essential for survival for a species to help them continue to adapt to new environments. Additionally, there can be non-harmful DNA mutations that simply change appearances. This could be anything from hair color, freckles, or even pattern baldness. A recent study published by the American Society for Microbiology concluded that when DNA is mismatched during repair in eukaryotic organisms, it contributes to a microevolution in a pathogenic fungus. Pathogens can survive in an outside environment, then enter in a human host and thrive. In the case of this fungal pathogen, it's Cryptococcus neoformans, and it was able to rapidly adapt due to a high frequency of DNA mutations. Ultimately, the study concluded that the hypothesis was correct. This pathogenic fungus had the ability to take advantage and cause disease via mutator phenotypes. As previously mentioned, sometimes errors occurring during DNA replication can lead to mutations that ultimately lead to cancer. One study by Tomasetti and Vogelstein proved that cancer incidences are correlated with the number of stem cell divisions in an organism's lifetime. The study tested a variety of types of cancer from people in 70 different countries. Very interesting study. It was found that nearly 2/3 of the cancer occurrences may be from DNA replication errors. Through the observations, it was clear that the results may vary due to external factors, such as environmental exposures and other internal factors like hormones or diet. The most common cancers developed from lifestyle habits include skin cancer from UV radiation, lung cancer coming from smoking, head and neck cancer from human papillomavirus, or HPV, and liver cancer from hepatitis C. Now, the common cancers developed from DNA errors are pancreatic, brain, intestinal, bone, thyroid, leukemia, ovarian, and testicular cancer. This can be frustrating for doctors who are trying to cure cancer, as DNA replication errors are part of a normal, healthy person's lifestyle as well, regardless of what you're trying to do to prevent cancer. Now, I will mention, although it is important to keep a healthy habit and keep doing those things that you know are good for you, cancer can be inevitable for some. And this further exemplifies why studying and understanding DNA mutations is so important. More recent studies are diving into some deeper factors that develop with these mutations. A study showed that defects in DNA repair genes are most commonly linked with cancers that are hereditary. It's estimated that with current cancers, only 5 to 10% are acquired from inherited abnormalities, meaning that the person was essentially born with a predisposition to cancer. The study further explains that alterations in mismatch repair, base excision repair, nucleotide excision repair, and double-stranded break repair structures show underlying cancer predispositions. As techniques for sequencing becoming more available, the likeliness for discovering occurrence of a germline mutation in tumors is increasing more than previously hypothesized. It is yet to be known if there are any clinical implications for the defects in DNA repair genes. It's amazing to think that just over 50 years ago, DNA itself was initially first discovered. Since then, many studies continue to be done to understand it further. With battling the negatives of DNA replication mutations, we can see that things like cancer can sometimes be inevitable. With that being said, we now know the importance of continuing these studies. At this point in time, I do want to encourage my students to take a look at these studies that were mentioned in this lesson. I want you to dive into these resources, further dive into the topic. The goal here is just to give a quick overview of what the DNA replication errors look like. And now that you have this basis, please take some time to get to use the knowledge you have now and keep expanding and building upon it. Thank you.